Maayong adlaw mga katusok, this is Faijah Marohomsalic of BSMLS4C and welcome to MedTech for the Day. In this video, I will be showing you different laboratory tests and procedures that are conducted in ISBB section of the laboratory. The first thing to do in every laboratory activity is to disinfect the working space as well as wearing the proper PPE. First is washing of RBC. This is done to remove the unwanted plasma proteins including antibodies that might interfere with the reactions. Step 1 is to remove the plasma from the red cell. Make sure not to disturb the bottom. Step 2, add as much volume of NSS until the mixture fills 2/3 of the test tube. Step 3, mix it by gentle inversion 3 to 5 times. Step 4, centrifuge the samples for 2 minutes at 3,400 RPM. Step 5, pipette and discard the supernatant. Make sure the packed red cells are not disturbed. Repeat steps 2 to 5 up to 3 times. On the final washing, the supernatant fluid must be clear. Next is red cell suspension preparation. 5% red cell suspension is a common reagent in many serological procedures. To do this, step 1 is to label 2 test tubes with 4 ml RCS. Step 2, add 200 UL of washed red cells to the labeled tubes. Step 3, add 3,800 UL of normal saline solution to the labeled tubes. Always remember, a proper red cell suspension has a tomato red color. Also, in preparation of red cell suspension, wash completely the given whole blood sample several times with NSS to remove traces of plasma. And lastly, the suspension should be free from any interfering substances. Mix it well, and now the red cell suspension is ready. Third is ABO and RH forward typing group using slide method. Step 1 is to label the slides with the patient's name. Step 2, add 1 drop of anti-A, 1 drop of anti-B, and 1 drop of anti-D on a clean labeled slide. Step 3, add 1 drop of 4% red cell suspension to each drop of reagent on the slide. Step 4, mix the reagents and the red cell thoroughly using a clean applicator stick for each reagent. Step 5, gently tilt the slide from side to side continuously for up to 2 minutes. Step 6, read, interpret, and record results of the reactions on each mixtures. Fourth is forward and reverse typing using tube method. First part is testing red cells. Label 3 tubes with anti-A, anti-B, and anti-D. Step 1, add 1 drop of each reagent on the corresponding test tube. Step 2, to each tube, add 1 drop of 4% suspension of red cells to be tested. Second part is testing serum or plasma. Label 2 tubes with A and B. Step 1, add 2 drops of plasma to the labeled tubes. Step 2, add 1 drop of A reagent red cell to the tube labeled A. Step 3, add 1 drop of B reagent red cells to the tube labeled B. Step 4, gently mix the contents of the tubes, then centrifuge for 15 seconds at 3,400 RPM. After centrifugation, read macroscopically with a direct light source for the presence or absence of agglutination. Agglutination of tested red cells is interpreted as positive test results. The fifth procedure is cross-matching using gel method. Step 1 is to label the tube. Step 2, dilute 10 UL donor's red cell with 200 UL DG gel sol to prepare 1% red cell suspension. Step 3, dispense 10 UL of donor's red cell suspension to the corresponding well. Step 4, dispense 25 UL of the patient's plasma into the same well. Make sure to label the well. Step 5, incubate for 15 minutes at 37 degrees Celsius. Step 6, after incubation, centrifuge for 9 minutes. And lastly, read and interpret the result. The sixth procedure is major cross-matching using the manual tube method. Step 1, mix 2 drops of patient's plasma with 1 drop of 4 ml donor's red cell suspension. Step 2, add 2 drops of LISS and mix. Step 3, incubate at 37 degrees Celsius for 10 minutes. Step 4, centrifuge for 15 minutes at 3,400 RPM and observe for hemolysis and agglutination. Step 5, wash the red cell 3 times with saline and completely decant the final wash. In this step, follow the steps that are presented in the washing of RBC. Step 6, after pre-washing, add 2 drops of AHG to the dry red cell bottom and mix well. Step 7, centrifuge for 15 seconds at 3,400 RPM and observe for agglutination. If there is no agglutination, record the result as compatible. Presence of agglutination means incompatible. Seventh procedure is antiglobulin test procedure. First is to label the tube. Second, add 1 drop of red cell suspension into a labeled tube. Third, wash the tube 3 times with normal saline solution. In this step, follow the procedures presented in washing the RBC. Fourth, completely decant the final wash and blot dry using a gauze or paper towel to remove residual saline. Fifth, add 1 drop of anti-human globulin to the cell suspension. Sixth, centrifuge for 30 seconds at 3,400 RPM. Examine the cell for agglutination, grade, and record. The eighth procedure is indirect antiglobulin test procedure. First step is to label the tube with proper labeling. Second step is to add 2 drops of patient's plasma to a clean labeled test tube. Third, add 1 drop of group O cell red cell suspension on the same tube. Fourth, add 2 drops of LISS and mix. Fifth is to incubate 10 to 15 minutes at 37 degrees Celsius. Sixth, centrifuge for 30 seconds at 3,400 RPM. After centrifugation, resuspend the cell by gently tipping. Seventh, wash the tube 3 times with normal saline solution. Follow the procedure presented in the red cell washing. Make sure to completely decant the final wash. Step 8, add 2 drops of AHG to the dry red cell bottom and mix. Repeat steps 4 and 5. Lastly, observe for agglutination, grade, and record. And for the last test is copper sulfate. This is done to determine the hemoglobin level of the donor. Step 1 is to massage the finger to promote blood flow. Step 2, disinfect with alcohol. Step 3, using a feather lancet or a pricker, prick the finger. After picking, wipe the first blood and allow the remaining blood to drop in the copper sulfate solution. Check whether the blood sinks or floats. After the test, apply patient care by putting on Band-Aid. The last step in every test procedure is to examine and record the results. As a medical technologist, it is our responsibility to release an accurate and reliable result to our patient. After conducting several procedures, we should dispose our waste properly and disinfect our working space. This is to promote cleanliness and safety in the laboratory. And of course, it should end with proper hand washing. Hand washing is important to prevent spreading infection and maintaining safety. This has been Faijah Marohomsalic, your MedTech for today. Thank you for watching.